The paper that I have chosen to present is on Ebola virus epidemiology, transmission, and evolution during 7 months in Sierra Leone by Park et al. First, I will just go over what Ebola is and its transmission. Ebola virus is a serious hemorrhagic inflammatory disease with a high mortality rate when left untreated. The first case was reported in 1976 in 2 locations simultaneously, in South Sudan and Democratic Republic of Congo near the Ebola River, where Ebola virus gets its name. The biggest outbreak since then has been in 2013, which happened in West Africa, with most cases being present in Guinea, Sierra Leone, and Liberia. The primary host of Ebola is bats, and the secondary hosts are other animals such as gorillas, chimps, and pigs. The virus is introduced to humans when they come into contact with infected bodily fluid. Often this contact is through secondary infected animals. Human-to-human transmission only occurs via direct contact with blood and other bodily fluids. It can also be transmitted via material that has the infected fluid in it. There is also a risk for sexual transmission, which is why patients affected by the virus are advised to get tested 3 months after negative test results. First of all, this study was carried out after the outbreak of 2013 to 2015, which was caused by the Ebola virus variant known as Mucuna variant in West Africa. This is the biggest outbreak to date with more than 11,000 deaths. The disease was first found at the end of 2013 in Guinea, and throughout 2014 to 2015 it spread to Sierra Leone, Liberia, and then the rest of the world, although only selected cases are found outside West Africa, and those were contracted by people working in West Africa. It is the only time where the disease has been sustained in humans for such a long time, which is why many studies were carried out to study genetics of Ebola virus to see what drives its longevity. Furthermore, the results from research will be valuable to global outbreak responses as it allows containment and treatment. This is a brief summary of the research paper. There were 232 samples collected over 7 months from patients from Sierra Leone. The research also included 86 previously researched genomes from earlier in the outbreak. The samples collected were only from patients that contracted the disease through human-to-human transmission. They used high-depth replicate sequencing to observe the host transmission and recurrent emergence on intra-host genetic variant. The study allowed the identification of many mutations in the Mucuna variant, which spanned throughout the outbreak. The results obtained from this study, which looked at the human-to-human transmission of the disease, will help in controlling human-to-human transmission, as well as the development of the candidate diagnosis and vaccine, as well as future therapies. High-depth sequencing was used in order to characterize the genetic diversity of Ebola virus within intrahost and between interhost. The samples were sequenced at 2 different locations, one in Sierra Leone, KGH, the Kenema Government Hospital, and in Bo CDC, the U.S. Center for Disease Control. They also performed a massively parallel genome sequencing from samples collected from Bo and Sierra Leone. Complete coding of 232 Mucuna variant was achieved in the results. They were found to be 18.5 kilobases in length. Despite extensive sequencing, full genome sequences were not obtained from the samples at KGH. This could be due to the country's deactivation of the virus sample, as well as other factors such as long delay in shipments. However, the success rates were much higher at the CDC, as only 7% of samples were not fully sequenced. This might be because the samples at CDC were more recently collected and were not lysed in buffer for long periods of time and were subjected to different deactivation protocols. The 232 and the previous 86 samples amounted to the total of 318 samples, which were analyzed and were found to obtain 464 single nucleotide polymorphism, 125 non-synonymous, 176 synonymous, and 163 non-coding. They also observed 5 single base insertion and 2 double base insertion in the non-coding regions. For cross-border contamination, previous study shows very little about the genetic diversity of Ebola virus in different locations. The first Ebola virus sequenced in Sierra Leone showed that it had stemmed from 2 distinctive lineages that had originated from Guinea. The first lineage, SL1, is more related to the variant found in Guinea than the second lineage, SL2, which was characterized by 4 additional mutations, which suggests that SL2 has evolved from SL1. A third lineage, SL3, which has been observed to be different from SL2 by a single mutation. SL3 has become the most prevalent variant in Sierra Leone in the first few weeks of the outbreak when SL3 appeared. It led to the disappearance of SL1 variant. This identification of SL3 is defining mutation, which is important as it is the first commonly circulating mutation observed to arise within the Sierra Leone borders. SL3 has also become the most prevalent lineage with no further development. Also, the samples show that 97% of the genome carry the SL3 mutation, with the rest of the group being SL2. This is illustrated in the following image. This phylogenetic tree shows the recently sequenced Ebola virus. The 232 samples were shown in dark blue. The image illustrates a high-resolution view of the accumulated genetic diversity between the different Mucuna variants. The results summarize all Sierra Leone Ebola virus cases to the initial introduction of Ebola virus into Sierra Leone. They also provide further evidence that all Ebola virus cases during the outbreak originated from human-to-human transmission rather than from a zoonotic introduction from the unknown Ebola virus reservoir. For human-to-human transmission, intrahost variants provide information regarding human-to-human transmission, such as shared ISNVs, has been used to estimate the relative size of the transmission bottleneck and to identify human-to-human transmission chains. In the dataset studied, it was found that several ISNVs are shared among 2 or more patients. The figure shows that the selection is not the primary case of the persistence, since synonymous and non-synonymous variants are equally common among shared ISNVs, and selective pressure is likely to be different for the 2 classes of the variants. The image outlines the samples spanning from 2013 to 2015. Variants shared between 2 or more samples are shown as rows of connected points, and each row is a genomic position. Each point indicates the presence of ISNV in the patient. From this image, we can conclude that a combination of human-to-human transmission and recurrent mutation is likely responsible for the ISNV pattern shown. In conclusion, we can see that human-to-human transmission occurred at the initial outbreak demonstrates very little genetic diversity within human hosts. However, the virus is able to thrive and replicate within a human host and accumulate vast genetic diversity. Furthermore, investigating genetic diversity will further drive future therapeutics and vaccine production. For further information, please check out the references provided.